So this is our fourth part in the DNA, the genetic material lecture. So again, keep in mind all the stuff we've talked about, part 1, part 2, part 3, the scientists involved in understanding the basis of DNA, the information, how it works, the basic replication process, the basic structure, all that from the first 3 parts. We left off in the last part talking about replication in prokaryotic cells. And the big thing to remember here is prokaryotic cells replicate from a single origin point. They literally just zip around that loop and create 2 copies. It's a very fast, very quick process. Some species, some types of bacteria or prokaryotes can do this in 20 minutes or less. When we look at humans, much longer process. We look at most multicellular organisms, it takes a lot longer. So there are slightly different enzymes associated with prokaryotic replication. E. coli bacteria uses 3 DNA polymerases. So a little bit different, but the same basic concepts. You're trying to take a strand of DNA, rip it open, add new letters to it, and ensure that you do it exactly correct. So there's still helicases involved, binding proteins, polymerases, ligases. We have the replication fork, the continuous strand versus the discontinuous strand. All that is very similar between prokaryotes and eukaryotes. So where we left off last time was looking at and talking about this thing called the replisome. This is a macromolecule that contains replication enzymes. So it's this crazy, kind of, I don't know what you call it, octopus-like structure that actually has all these different pieces and parts that enable prokaryotic cells to replicate very rapidly. The goal is identical copies when these copies are being produced. But we all know mistakes happen. It will occasionally mess up, and this creates new variations and new strains of bacteria. So here's kind of the overview of how it works. Step 1, step 2, step 3, step 4, step 5. I'm not asking you guys to know the intricate details here. You'll get this later, again, when you move into a higher level genetics course, molecular biology, those types of courses. But just get comfortable with the basics of how it works, the similarities between prokaryotic and eukaryotic replication, and similarities, but then also differences. Okay? All right, so with eukaryotic replication, this would be us, as well as plants and fungi and protozoans. We have larger amounts of DNA located in multiple chromosomes that we have to replicate. Okay, so here's the general picture of the DNA. You've got your double helix, it wraps around all those little histone proteins. That's what the video was showing. Then it coils and coils and coils and coils and coils and coils into these little things called chromosomes. And then in metaphase, they're all lined up in the center of the cell. So there's a larger volume of information that's being copied here, that's being replicated in eukaryotic cells. So there are some issues or some things of concern to try to ensure that it happens correctly. So one of the things we'll see with eukaryotic cells is that when they replicate, these things called telomeres are associated. Now, telomeres are these little caps that go on the end of the chromosome. So right over here, this is a telomere. It's kind of like putting a little hat or a little sock on the chromosome. Telomerase is the enzyme that actually builds the telomeres. Telomeres have been identified with cell aging. So this blank here is, telomeres play a role in cell aging. Every time the cell replicates, the telomere cap moves up a little bit. And basically it chops off a little bit of the chromosome, and then another one, and then another piece, and another piece, and another piece. So if you look at the diagram down here, it literally is shortening the chromosome every replication cycle. It's approximately 50 cycles before the cell dies. And it's believed the telomeres just shorten it up so much that there's not enough functional material there, causing it to lead to cell death. Now what's interesting is cancer cells, we talked about that in the last unit, cancer cells do not shorten up their telomeres. They don't move up that DNA strand or that chromosome, and that might be why those cells never die. Cancer does not stop dividing. It does not die. That's a huge area of research right now, trying to understand why cancer cells will not die after 50 cycles. So our skin cell, 50 cycles and it dies. Our hair, our bone, our muscle, etc., roughly 50 cycles and those cells die. So a lot of times when we're looking at cells and they're going through the replication process, we try to keep an eye on things that can cause mistakes, these errors in replication. So there's a lot of factors that can disrupt the replication process. There are things like UV radiation, solar radiation from the sun, X-rays, different chemicals. Are we using things to preserve our food? Nitrates and nitrites are preservatives used in processing meats. Those have been known to cause mutations or changes to the cells. Cigarette smoke, things we call carcinogens, play a big role in this. Benzoyl peroxide, this is a common ingredient in acne products. That has been known to cause problems with cells going through the cell cycle. So what we want to keep in mind are things called mutagens. These are different, I want you guys to fill in the blanks here. Mutagens are different substances that increase the number of mistakes or mutations that can occur in the cell. And what they're doing is they're disrupting or messing up the replication process. So the DNA is not copying correctly. Now, fortunately, we have advantages, or we have things that can go in and repair the DNA. And we usually see DNA repair occurring in two ways: specific repair and non-specific. So the specific repair is where the DNA will go in and actually identify a very selective spot that is a problem and try to fix just that spot. It goes in and says, oh, this piece of DNA is incorrect, it's not right, it's got a problem, let's fix that. And it tries to specifically repair a certain location of the DNA. Non-specific repair is when there's a single mechanism that goes in and just tries to fix everything that's wrong. Kind of your handyman that goes in and corrects all the problems in the house. Specific repair is I'm bringing in the furnace guy to fix just the furnace. So DNA does this. It tries to repair itself. That's part of the checkpoint. That's part of the job of ligase, part of the job of the structures associated with the DNA replication process. Can they go in and fix any of these mistakes? So hopefully they're always fixing any errors that occur, but unfortunately we know mistakes do get through. So damage that can occur from sunlight or UV light can cause problems. Or actually, let me reverse that or say it in a different way that makes more sense. UV light can cause damage to a DNA strand. And what it does is it causes these two thymines to actually bond together when they should not. So instead of a T and an A, you have a T and a T. Not good. Not the way it should work. But what we'll see is photolysis is produced by our exposure to sunlight. That actually is an enzyme that will break that thymine-thymine bond. So we see this as a type of repair called photorepair. So some level of exposure to the sun is actually healthy, is good for us. It enables our body to produce photolysis because of exposure to sunlight, and that will potentially break up these thymine-thymine bonds that occur as a result of exposure to ultraviolet radiation. So there are these natural mechanisms built into our body that is hopefully trying to correct mistakes in DNA. So here's the example of what happens when UV light is present. It causes this distortion of the thymines, causing two thymines to bond. Exposure to sunlight produces photolysis, which binds to the damaged DNA, corrects it, basically pops those two guys apart, so they've been locked together at the bottom there. pops them apart, allows them to bond back to the adenines that they should be bonded to, and hopefully correct the mistake. So that's one mechanism where we can see repair happening. And another general mechanism is this thing called excision repair. This is a non-specific. This is when certain various types of enzymes come in and they recognize the damaged DNA, and they literally go in and cut out the bad piece and tell polymerase, hey, go glue in new pieces of DNA. Go glue in and repair that stretch that's been messed up. Let's get the correct letters in there and fix the problem. So all in all, as a cell spins through the cell cycle, it is continuously going to have problems, mistakes, errors. That's just natural. But hopefully all those preventative mechanisms are in place to fix those mistakes, repair the damage, and try to ensure our DNA stays healthy, functional, and doesn't create long-term problems for us.